Okay, welcome to Chapter 14. In Chapter 13, we looked at control of microbial growth on surfaces, whether it was an inanimate surface or a living surface. Chapter 14 is going to look at antimicrobial drugs. So antibiotics, antivirals, antifungals, various drugs that are used to treat actual infections. And I think for most of us, we're finally getting to the really interesting part of microbiology. For Chapter 13 and 14, I provided that table that listed for Chapter 13 the types of physical and chemical controls that you need to know how they act and examples. For Chapter 14, there is a table for the targets for antimicrobial drugs. That's not going to be all you need to know from Chapter 14, but it will be the way to organize these are the drugs that you need to know from this chapter. Move myself out of the way here. There we go. Antibiotics are a relatively new thing. They were first mass-produced in the 1940s. So my parents were of the generation that were the first to have any access to antibiotics. They were seen as a silver bullet, be able to use for pretty much anything, and they can be used for many things, but the overprescription of antibiotics does have real problems. We have the evolution of drug-resistant pathogens. And we'll cover an example of evolution of antibiotic resistance in real time later in this lecture. So, synthetic antibiotics, which are the primary types of antibiotics used today, are not the only antimicrobial drugs. There's certainly herbalists and herbs that can be used, but they're not the most common type of antimicrobial. And for the purposes of this class and my entire lack of knowledge of Chinese herbs, we're going to focus on the commercially available antibiotics, antivirals, etc. Again, you're not going to need to know the names of the scientists, but one of the first antimicrobial agents was what he called compound 606 that treated syphilis. That is a sexually transmitted illness, but it can have systemic-wide effects, including bone-deep sores and mental health issues. And we will cover our sexually transmitted illnesses in a later chapter. Fleming is the one that discovered naturally produced penicillin. Penicillin comes from Penicillium mold. Petri dishes got contaminated and the mold killed the bacteria, so that was a nice accidental discovery. And then later we were able to determine how to scale up that production, so we're not just trying to extract it from mold spores on a Petri dish. And finally, we just have an example of a scientist getting antimicrobial productions out of soil bacteria, scaling up the volume of microbes that can be grown and perfecting the purification process. This is showing us what can happen if we overuse or misuse antibiotics. There is a term called the spectrum of activity. So how broad the spectrum, the range of microbes against which an antimicrobial works. So a broad spectrum antimicrobial means it kills a lot of different things. A narrow spectrum antimicrobial will kill only one specific microbe or only one category of microbes. This slide is showing us what can happen with the overuse of a broad spectrum antimicrobial. So we have a mixture of organisms, including things that are part of the normal microbiota, the normal flora, the normal biome, all those microbes that are supposed to be living in and on us. And we have a minority of the cells are this little blue-green pathogen. A broad spectrum antibiotic will kill everything that's sensitive to it, and if this one little pathogen happened to be resistant, now there is no competition in its environment, and all of the food, all of the space, all of the water is there for this antibiotic, this drug-resistant microbe, to take over. This is the sort of thing that happens with C. diff, the Clostridium difficile infection. The person's normal flora is present along with the offending Clostridium. Repeated use of antibiotics that may truly have been necessary for other infections eliminated the normal flora in their intestines, and then the Clostridium difficile proliferates and causes a super infection. This is our evolution of antibiotic resistance in real time. So initially, of course, the microbes are growing just fine where there's no antibiotic. That allows, in this experiment, to make sure that you have a viable, healthy population of E. coli. This edge here on each side is where this population is going to encounter the next band on that giant table-length petri dish that has tenfold the amount of antibiotics that E. coli should be able to resist. This antibiotic should be very effective against them. E. coli do have flagella. They are very good swimmers, and because they're really thick here, they are going to swim toward the interior of that giant petri dish to get toward where there's more nutrients, more space, more water. The fact that there's a high level of antibiotics means that there aren't going to be many of them that will survive, but the few that do won't have much competition, and they will be able to proliferate. "11 days. In 11 days, we went from completely susceptible E. coli to E. coli that are rapidly multiplying and can withstand 1,000-fold the amount of antibiotic that these original cells could withstand. Pretty impressive. So 11 days to go from completely sensitive to an antibiotic to resistant to 1,000-fold. And considering that the generation time, the doubling time for E. coli is 20 minutes, we can get an awful lot of bacteria in a very short period of time. And if those E. coli are not constrained to a tabletop in a lab, but are in a patient or in some environment, then they also have the capacity to pass that antibiotic resistance on to other bacteria in the environment through transformation, through transduction, through conjugation. This is showing us the various concentrations of a drug in a patient depending on how it is administered. Most antibiotics we get, most antimicrobials we take are oral, meaning we ingest them. We can also have intramuscular injection of an antimicrobial, or we can have, if it's a truly serious case, intravenous. The intravenous, we get enormous concentration of the drug in the patient, in the plasma, very, very quickly, and then it falls off pretty quickly. An intramuscular injection takes a little longer to peak, but it also takes longer to decline. And oral antibiotics take much longer to peak and then stay in the system a little longer. This is why with oral antimicrobials, it is really important that we take the entire prescribed course of the drug. And it's difficult to do because typically we feel better after a couple of days. And when you feel better, you're not as motivated to take your medication. And it's easy to forget to take the oral doses of an antimicrobial. That is why if there is an injectable available for an antibiotic that I truly require, I will take that because it's one time. You get the intramuscular dose and that is it. You have completed your antibiotic course. Otherwise, I set an alarm in my phone for the right number of days so that I will remember to take all of my drugs. This little cartoon is showing a generic prokaryotic cell, and each of those yellow boxes indicates a target for antibacterial compounds. And antibiotics are against bacteria. An antibiotic won't work against a virus or a fungus or a worm. It will work against bacteria. These targets are such that they are different from that chemical or structure in the host, and we're being very egocentric here. We are the host, right? If we were in a veterinary class, there'd be different hosts. But we're going to be talking about the host is humans. The pathogen for this part is bacteria. So the table that I produced for you has these different targets on it for you to look at the different ways those targets are affected. So if we start at the top and work our way clockwise, the plasma membrane, that's a tough one because plasma membranes are really similar between prokaryotes and eukaryotes, but we have cholesterol in our plasma membrane and there's a different sterol in the plasma membranes of prokaryotes. So polymyxins can attack the plasma membrane of the prokaryotes with little side effects against us. And we can also use a lipopeptide called daptomycin. If we are attacking the ribosomes, we are inhibiting protein synthesis. And the ribosomes in prokaryotes are structured similarly. There's a small subunit and a large subunit, but they contain completely different proteins and different RNAs. And that's why we can target prokaryotic ribosomes without affecting the ribosomes in the human host cells. Two different drugs target the small subunit, a category of drugs called aminoglycosides, and we'll see examples of those, and tetracyclines. They are called tetracyclines because cycline, they have 4 circles, and tetra is 4. Drugs that target the large subunit are the categories of macrolides lincosamides, chloramphenicol, and oxazolidinones. It's a tongue twister. Metabolic pathways are also tricky. The way glycolysis works in a prokaryotic cell is exactly the way glycolysis works in a eukaryotic cell, and for much of the rest of cellular respiration. However, the ways that prokaryotes synthesize their folic acid is different from us, so we can target the enzymes in folic acid synthesis. And those drugs are the sulfonamides, sulfones, and trimethoprim. We can also inhibit mycolic acid synthesis in the mycobacteria, things like Mycobacterium tuberculosis and Mycobacterium leprae. And the drug example here that inhibits mycolic acid synthesis is isoniazid. DNA synthesis is really tricky. The enzymes are extremely similar. They're not identical, but they are similar. So there tend to be some fairly toxic side effects when using drugs that target nucleic acid synthesis. But fluoroquinolones like Cipro and Levo and moxilofloxacin inhibit DNA synthesis, and RNA synthesis is inhibited by rifampin. So these drugs are used if there aren't any other options, but they do have some fairly nasty side effects for the host. The cell wall, however, is a very decent target. We don't have cell walls. The cell wall of prokaryotes, at least all the ones that are pathogenic to humans, have peptidoglycan. There is no peptidoglycan anywhere in a human cell. So we have a category of drugs called beta-lactams. We're going to see their structure in a bit. They have a ring called a beta-lactam ring. So penicillins are beta-lactams, so are cephalosporins, monobactams, and carbapenems. And there are some glycopeptides that attack the cell wall, like vancomycin. And then we have a topical antibiotic called bacitracin that targets the cell wall. That doesn't mean there are no side effects of taking antibiotics that affect the cell wall. All of these could disrupt the normal flora, but they're not going to be harming the host cells. Not going to show you the drug, and you have to tell me which drug it is, but this is showing you what that beta-lactam ring is. It's funky. It's actually square. I don't think we've seen any other chemical so far that is square. So the beta-lactam ring is what is shared in all of the beta-lactam drugs. So penicillin G particularly is active against gram-positives. That's what this G means. Ampicillin affects a few more gram-negatives than penicillin does. But typically, because we are disrupting the cell wall, drugs that target the cell wall are more effective against gram-positives than against gram-negatives. And that seems a little counterintuitive. Gram-negatives have a thin cell wall. Why don't drugs that attack a thin cell wall damage gram-negatives at least as well as gram-positives, if not better than it attacks gram-positives? Well, that is because the cell wall is thin. That thin cell wall doesn't rely on multiple layers to be the correct cell wall and protect all the functions inside that cell. So if there's only one layer of peptidoglycan, the gram-negative is like, okay, that's fine. But if a gram-positive cannot get those multiple layers of peptidoglycan, its cell wall isn't going to function, its transport is not going to be correct, and that cell will die. And the route of transmission is listed for these. You won't have to memorize which way these drugs are taken. But parenteral means injected. The drugs that affect protein synthesis are going to be acting primarily on the ribosome. So chloramphenicals are going to be binding to the 50S subunit, they prevent the peptide bond formation. So if the organism cannot link its amino acids to build the peptide chain that then gets folded to make protein, it's not going to survive. It's going to have correct protein synthesis to survive. So chloramphenicol and the macrolides disrupt that peptide bond formation. Aminoglycosides bind to the small subunit. And that means that we can't do proofreading. It doesn't correctly bind to and direct the tRNA coming in to the codons. It causes that little mRNA to bend, and it's not going to work correctly. The tetracyclines also bind to the 30S subunit, but it prevents the tRNAs from binding at all. And if tRNAs cannot hydrogen bond to the codons, then we can't get any protein synthesis happening. Sulfonamides and trimethoprim are called anti-metabolites. So these drugs would go on that chart under drugs that affect metabolism because they are anti-metabolites. They block metabolism. And these two drugs are doing so by interfering with the folic acid synthesis that's done in a different pathway than how we do it. So the enzyme should be able to convert the PABA into the, sorry, my brain just stuck, into the drugs that we need, the folic acid that we're looking for. So the sulfonamide blocks, it is a structural analog of the PABA, the premetabolite for folic acid. Sulfonamide looks a lot like it. This ring structure is identical, but this functional group is different. And when that drug is used, when sulfonamides are used, the entire pathway of folic acid synthesis is stopped. The trimethoprim blocks the second stage of the synthesis, and we still don't get the final folic acid. And the reason why cells need folic acid is when we have the complete pathway, we convert the PABA into dihydrofolic acid to tetrafolic acid, eventually to folic acid, and that gets used to make nucleotides. So if we block this metabolism to make folic acid, we are also blocking the organism's ability to do DNA synthesis. This is why folic acid is one of the prenatal supplements that are given, because during development from an embryo to a fetus, there is tremendous cell division, and cell division depends on the presence of nucleotides. So making sure that there's plenty of folic acid in the diet of the mother means that the growing fetus also has plenty of folic acid that then can be used for building nucleotides. We use a different pathway, so blocking folic acid synthesis is not a problem. We don't synthesize folic acid, we eat folic acid. So blocking the synthesis of folic acid with sulfonamides and trimethoprim is an effective antimicrobial choice. This is showing two different sterols, cholesterol, which is found in our cell membranes, and ergosterol, which is found in fungal cell membranes. They're very similar, but if we have a drug that is specific for ergosterol, then we have a good antifungal medication. So the antifungal drugs, we can use flucytosine that inhibits DNA and RNA synthesis. We can disrupt microtubule function with a drug called griseofulvin. This is a drug that definitely would have to be topical. for a surface fungal infection. We would not want to inject or ingest Grisial fulvin because we don't want to disrupt our microtubules. Then we cannot do cell division. We can't support the internal structures of our cells. Likewise, drugs that inhibit mitochondrial function, would not want to ingest those. But we can disrupt the membrane with polyenes. We can inhibit ergosterol, and those will be very specific to fungal cells. We can inhibit chitin synthesis. Chitin is the cell wall of fungi. We don't have chitin. So polyoxins and nicomycins block chitin synthesis. And we can also inhibit these sugars called beta-1,3-glucans. We don't have these structures in us at all. Now we're going to look at some antivirals. Acyclovir is used for treating herpes virus infections. That's at least one of the infections it's used for. It is called an analog of guanosine. This is a guanosine. It would be a nucleotide if we had phosphates on it. But there is the ribose and there is the guanine. So acyclovir has the same two-ring structure as guanosine, but it doesn't have the ribose. It is without the circle. That is why it's acyclovir. And because it is similarly shaped to a nucleotide, it has the guanine base. It gets put into the viral DNA when it's replicating, and when the acyclovir lands instead of a guanine nucleotide, that stops the DNA replication. So acyclovir interferes with the viral DNA replication. There's also something called antiretroviral therapy. If you think way back to our chapter on acellular pathogens and viruses, we talked a little bit about retroviruses. Those are viruses that have an RNA genome that uses reverse transcriptase delivered by the virus to make a DNA copy of the viral RNA genome, and then that viral DNA gets into our cells and sticks into our DNA. The one retrovirus you've probably heard the most about is HIV, human immunodeficiency virus. The antiretroviral therapy that is used, we have a couple. We have drugs that will inhibit that reverse transcriptase. Those are very effective. AZT and etrovine are the ones that do that. If you can block the action of that enzyme, then the virus cannot get a DNA copy of its genome. If it cannot get a DNA copy of its genome, it cannot stick into our chromosomes. We also have integrase inhibitors. In order to get the viral DNA into the chromosome, requires the viral protein called integrase because it allows DNA to integrate into our chromosome. If we block that, even though the viral DNA got made, if it cannot get into the host chromosome, then it won't get transcribed or translated. We have fusion inhibitors that prevent the virus from sticking to the host and getting in. We also have protease inhibitors. In order for the virus proteins to be processed and end up to be their proper 3D shape, they rely on a chemical called protease. That is an enzyme that acts on the proteins to get them in their final proper shapes. If we inhibit the protease, then the virus can't assemble. So we have several targets for anti-HIV drugs. We can target the RNA by blocking the reverse transcriptase. We can block the DNA getting into our cells by blocking integrase. We can block the proteins from forming with protease inhibitors, and we can prevent the fusion of the virus with the cell surface. And an antiviral discussion would not be complete without remdesivir. This is a drug that was developed for treating other viral infections, but is showing some promise in treating that should be SARS-CoV-2. Bad typing, I'll have to correct that. SARS-CoV-2. COVID. Have a patient that gets intravenous remdesivir. The red blob is the initial drug that is injected. During metabolism, it is converted to its active form. So the green dot is the active remdesivir in the patient's bloodstream. This is the SARS virus that causes COVID-19. It is an enveloped virus, so when it binds to an ACE2 receptor on our cells, the whole thing gets taken in, and its RNA genome is released into our cells. Normally, the ribosomes would translate that into viral proteins. And then we also need, once that is done, we get this protein called RD, RP, which is RNA-dependent RNA polymerase. In cells, RNA polymerase binds to DNA and transcribes the DNA to RNA. And RNA-dependent RNA polymerase does not happen in cells. This is only viral. So the virus delivers its genome. Our ribosomes read it and translate it to make this polymerase. This polymerase would normally read the RNA and make more RNA, but remdesivir binds to that polymerase and blocks viral RNA duplication. And that is the mechanism of the remdesivir. This is showing us how microbes can become resistant. First part of this was how do we get antimicrobials to eliminate microbes. Now the microbes laugh at us and say, not today. So we can prevent the drug from entering. So beta-lactams, tetracyclines, and fluoroquinolones, bacteria have ways of blocking their entry. They're large molecules. They can develop ways of blocking the entry of those drugs. They can modify their targets. Slight changes in which protein or which RNAs are in the ribosomes will block the drugs that act on the ribosomes. The DNA, we're not going to mutate it, but it can fold differently. It can have proteins bound to it, and then that means the drugs that target ribosomes or that target the DNA can no longer act. There can be inactivation. There are enzymes in the bacteria that just say, all right, the drug got in, the target is fine, but the enzyme is just going to inactivate that drug. And lastly, there are some organisms that can just pump the drug back out. How do we determine how much of a drug is going to actually be effective? We start by testing it in test tubes with varying concentrations of the drug against a standard concentration of bacteria. Once we have an idea of that, it gets moved into animal models. Once it goes through animal models, it goes into people models, and once it goes through people models, it gets approved. This is called the dilution test. These tubes have, from a very low concentration of 2 micrograms per milliliter of the drug, all the way up to 32 micrograms per milliliter. And then each of these tubes was inoculated with a standard concentration of bacteria. So the exact same number of live cells was put into each of those tubes. We incubated overnight, and what it looks like is that 8 milligrams per mil is the minimum concentration that inhibits microbial growth, because we don't see any turbidity. The next step would be to take a loop from that tube and streak it onto a plate and see if there's anything in there. But this at least is the minimum concentration to inhibit, the MIC, M-I-C, minimum inhibitory concentration. This is just a way to do a whole bunch of that on a smaller scale. This is a plastic plate that has 96 little wells in it. And so we vary the concentration from low to high, going across the plate. And going down the plate, we have different drugs, and then we can put the same bacterium in same concentration of the same bacterium in all the wells, and then we can see here it looks like clindamycin doesn't really do too much. It looks like the minimum inhibitory concentration is higher than what we have in this dish. For this second antibiotic, as soon as we get to this concentration, a much lower concentration was effective against the bacteria. This one, even more so. Here we're showing with penicillin, once we got to 0.06 micrograms per mil, we inhibited. And then the last 2 wells is a well that just has no antibiotic and has bacteria, just to show that an active culture was used. And of course the last one is the uninoculated control. This is a strip of paper. The numbers on it represent, at that line, that is the concentration of drug. And this is VA, I believe that's vancomycin. So the Petri dish was completely slathered with a known concentration of bacteria. This strip was laid down on it, and then the plate was allowed to incubate. The drug will diffuse evenly out from that strip. And so what this shows is until we get to around 1.5, probably micrograms per mil, no effect whatsoever. We get to 2, and we're mostly killing it. There's just a few little dots. By the time we get to 6, where the drug is diffused, we've got good killing. The drug has diffused past where this clearing is, but the further it gets from the strip, the more dilute it is. And then once we get up here, we've got very effective. These lines are the writing on the bottom of the dish. There is no growth in this zone. This slide is showing us the number of new antimicrobials that have been approved up until 2012. So the 80s looked like a heyday of antimicrobial development, but in recent years, we had two new ones between 2008 and 2012. That's pretty sad. Again, you have that table to fill in for Chapter 14. That is not all you need to know from the chapter, but it is the drugs you need to know. So you can look at the images and match where the targets are to find out which drugs are there, and within each category, there might be different parts of that category that are affected. Like the cell wall, some of the drugs inhibit cell wall synthesis. Some of the drugs inhibit cross-linking. So you have to match and make sure you have the right drug for each target. And as we did for the first and second exams, for the third exam, there will be a study guide that will provide the rest of the information that you need to know. A good idea for getting a concept of what will be on that study guide is to look at the learning objectives that are at the top of each section of your textbook.